Hi, my name is Jack Rogers, and I'm going to be presenting on the Bioinformatics add-on for the Orange Data Mining Suite. So today we're going to be looking at this Bioinformatics add-on and some of the nice little tools it has in it. Now, this is really useful for anyone who does bioinformatic research and has been playing around with some of the machine learning algorithms for classifying different types of data. So Orange is useful for any type of real data mining, but for these purposes we'll be looking at how the bioinformatics add-on has some nice tools to make dealing with these sort of high-dimensional data sets more easy. So if you don't have the bioinformatics add-on, you'd want to go up to Options, Add-ons, and sure enough, there's a little bioinformatics add-on right here. It may prompt you to refresh the lists of your repositories, which you can just hit OK, and then this little bioinformatics thing will pop right up, and you can click this little checkbox if it's not already clicked. So to start off, we'll be looking at this GeoDatasets widget, which is conveniently placed right up here. So what does this do? Well, this gives you access to about 2,600 different data sets that you can just click on one and hit commit, and it will download. And if you've ever used Orange before, if you have a file that you want to load in, you can just bring your file widget down and then select what file you want to use. And there's an example of some bioinformatic data that we've been playing with lately. But in this example, we're going to assume that we don't have any files downloaded, but we still want to do bioinformatic research. So the GeoDatasets widget makes this really easy. So we can select anything in here and hit commit, and it will work in the same way our little file widget does. So in this example, we're going to use this alternative splicing of 5 different tissues from human samples. It's looking at brain tissue, muscle tissue, liver tissue, testes tissue, and bone marrow tissue. And this is data comparing the mRNA expression levels of various genes of interest that help differentiate those tissue types. So to give you an example of what this data looks like, here it is. So what we have is 9 samples from the expression level of 391 different genes of interest. So for example, let's go to the end here, and we see that with this little class column we can tell which row is associated with which different tissue type, and then we can see the mRNA expression levels for each of the genes, and we can tell, just look at SNRPF, we can tell that between liver tissue and brain tissue, liver tissue tends to express this gene to a much greater extent than these brain tissue samples. So I can't tell you anything interesting about that offhand, but if you're a bioinformatic researcher, little things like this are why you're actually playing with this data. So this is how we look at the data with by samples with genes as our column headers, but what if we wanted to transpose this matrix? Well, the GeoDatasets widget makes that easy. So we can choose to transpose our output based on genes or samples. We were looking at samples before, which means that each different row is an individual tissue sample. So if we look at it from the perspective of genes, and we hit commit, then all of a sudden when we look at our data table, this will have changed. The genes will actually be our rows, and the columns will be the actual tissue samples that we're looking at. So now let's look at another GeoDatasets example. This is medulloblastoma metastasis. So this is an example of 23 different tissue samples from people with and without, or for all people with medulloblastoma tissues, 13 of which are non-metastatic and 10 of which are metastatic. And if you're not familiar with that, I believe that has to do with the malignancy of the genes, or of the tumors. So it's very important in the field of bioinformatics to determine what are the genes of interest that decide or help decide whether a tumor is going to be deadly for the patient with it, or what is it about this tumor, or what is the biological indicator that allows us to know if this tumor is more dangerous or of a different type which can lead to different approaches for kind of personalized treatment in the medicine field. So we can see that we can download a wide array of different types of data with this GeoDataSets widget. And if you've ever played with bioinformatics data before, one of the biggest problems is data parsing. Science labs all across America and the world have a lot of different type of standards and formats that they like to put their files in, and it usually becomes a problem where anytime you'll get a data set, you'll have to go through and you'll have to parse it in a way specific to that data file. GeoDatasets is one of the few places that I've found where you can literally just plug and play high-level bioinformatic data, and because we're in Orange, we already have a large amount of high-level tools to begin analyzing this data. So back up to our tissue splicing examples. Let's take a look at the volcano plot widget, which is used for determining genes of interest. So I won't claim to be an expert on what a volcano plot is. I believe it's to help you find genes of statistical significance or outliers in your data that may be worth looking at more heavily. So the idea is that significance goes up on the y-axis, and the idea is that things inside of these red boxes in the corners are the most significant samples. So in this data, at least under the default parameters, we don't have anything that looks extremely significant, but there's a lot that we can do with this. For example, let's say we wanted all of our liver examples, so we can just click on liver and we can hit commit, and then if we bring down another data table, when we take the output of the volcano plot and send it to a data table, and it doesn't work. So bad example. Let's just forget I said anything about that. I apparently need to play with volcano plot a little more. But this is an example of how we can just out of the box, straight from getting our data input, use this bioinformatic widget to determine some statistics, to visualize our data in different ways and possibly look for some interesting things of statistical significance. But I'm not an expert on volcano plot. You can go to Wikipedia and there's a very nice article on it that I have not yet mastered. But the problem with this whole bioinformatics on is there isn't actually any documentation for any of these widgets. So this should be one of the first examples of that. And I guess it won't be the highest level, at least in the case of volcano plot. But anywho, moving on, let's take a look at this gene info widget. Now this one's really useful. So we have in our data set here, this is the tissue sample data set, we have how many genes? We have 392 genes. So if we're looking at all these genes, we want to figure out what do they mean? What do they do? Well, luckily enough, this bioinformatics add-on contains the gene info widget, which if we have a data set with genes in it, and the genes are labeled with their actual gene name, then we can just draw a little line over to this gene info widget, and sure enough, we have a list of all these 392 genes and the descriptions, and we can get some links to their pages on the NCBI webpage. So this is great. We're looking at a gene and it's showing itself to be statistically significant, and rather than go and Google it, we can just stay right inside our platform and look at what this gene does and determine if that, in our mind, is part of some sort of molecular pathway that might be of interest in looking into further in a lab setting. So the idea is we're doing all this bioinformatic research, we're looking at all these genes, we're trying to figure out what's causing these diseases or these difference in tissue types, etc. This gene info widget makes it much faster for us to go through and identify genes of interest by determining why these genes are getting the expression levels they are in the context of our problem. So now we're going to look at our data profiles widget. So this is looking at the expression levels of the 9 different samples that we have in our differentiated tissue sample data set that we got for the geodataSets widget. So this is looking at the individual samples, and these are their error bars as far as the kind of mean expression levels of the different genes, and we can see that there's pretty high error in all of these because, you know, when you're looking at 300 and on the order of 300 genes, and they're all kind of, you know, randomly expressed, they're all not part of the same kind of metabolic pathway, there's a very large chance that you're not going to be able to find, they're not all going to be high and they're not all going to be low. These are not all genes with the same sort of molecular mechanism, at least in this example. So this is an example of where we might want to transpose our data set. So go ahead and transpose that, and hopefully it won't take too long. Now we've already got this data set downloaded, so I won't be very upset if it takes a long time. But sure enough, how about that? Ah, the glory of orange.